Hello and welcome to Bio40 Pre-Lecture 20. Today's topic is translation. We talked last time about how mRNA is transcribed from DNA. Now it's time to discuss how the mRNA is translated into a polypeptide chain. We're going to divide this concept into two parts. First, we'll cover tRNAs. We talked about them briefly a couple lectures ago. They are the adapter molecules that connect mRNA codons to the appropriate amino acid. Then we'll discuss how ribosomes mediate tRNA-mRNA interactions and build polypeptide chains. This is an example of a tRNA or transfer RNA. They are short, single-stranded RNA molecules a little under 100 nucleotides in length. In eukaryotes, they are transcribed by RNA polymerase III. But in addition to the regular RNA bases, tRNAs have several modified bases, which we'll discuss shortly. Each tRNA is attached to, or charged with, an amino acid. For instance, this is an alanine tRNA. Let's take a closer look at tRNA structure. Unlike DNA, RNAs can have more complex structures. The primary structure of a tRNA is its series of bases. tRNAs have several sequences that pair within the strand, forming this cloverleaf secondary structure with multiple stem loops. These loops fold and form an L-shaped, three-dimensional, or tertiary structure. We'll mostly just show the secondary structure for ease of viewing. The hydrogen bonding that forms the stem loops are generally the normal GC and AU pairs, but within a single strand. The amino acid is attached to the 3' end of the tRNA. So again, this is an alanine tRNA. At the bottom of the image, labeled with the Roman numeral 2 here, is the anticodon loop. The 3 bases in yellow are the anticodon, which will base pair with an alanine codon. The other parts of the tRNA have to do with charging, or making sure the right amino acid is attached. I mentioned before that tRNAs have modified bases, and by now you've probably noticed letters besides A, C, G, and U. One important modified base is inosine, which is demethylated adenine. You can see that inosine is part of this anticodon. There's also methylguanosine, lysidine, which is a cytosine modification, and several different uracil modifications. We'll discuss some of their roles later in the lecture. Now let's talk about how the tRNA molecules are charged with amino acids. The last 3 nucleotides on the 3' end of the tRNA are always cytosine, cytosine, adenine, and the carboxyl of the amino acid is covalently bound to the 2' or 3' hydroxyl of the sugar of the last base. Ensuring the right amino acid is paired with a given anticodon is key for proper translation. The charging is done by enzymes called aminoacyl tRNA synthetases. There are at least 20 of them in any given organism, one for each amino acid. Each aminoacyl tRNA synthetase recognizes a tRNA molecule and attaches the appropriate amino acid. Sometimes there are multiple tRNAs that are used for the same amino acid, so a given enzyme could charge more than one tRNA. These are the true molecular translators of the cell. They recognize a specific nucleotide sequence and make sure the right amino acid for it is attached. This diagram shows how charging works. Aminoacyl tRNA synthetases have several binding pockets. One recognizes a specific amino acid, while another recognizes the tRNA that corresponds to that amino acid. For instance, here you see a glycine in one binding pocket and a glycine tRNA in another pocket. And like in so many other processes, the energy here is provided by ATP. In the first step of the process, the triphosphate bond of ATP is cleaved, and AMP, that's adenosine monophosphate, is attached to the carboxyl carbon of the amino acid. In the second step, the carboxyl carbon is attached to one of the hydroxyl groups on the last ribose sugar of the tRNA chain. The tRNA is now charged. Now when the anticodon of the charged tRNA base pairs with an mRNA codon, this indicates that the amino acid attached to the tRNA should be the next amino acid in the polypeptide chain. Researchers were able to demonstrate this by fooling in vitro protein synthesis systems. By treating tRNAs with certain chemicals, they could replace the amino acid attached to a tRNA without altering the codon or anticodon. So if you attached alanines to cysteine tRNAs, then added an mRNA that had cysteine codons, the in vitro system would put alanines in the polypeptide. Let's talk about these anticodons. Sometimes the pairing of codons and anticodons is very simple, as in this example here. Read 5' to 3', GGA is a glycine codon. So CCU, read 3' to 5', would be a good tRNA anticodon to pair with this codon. But is this the most economical way to do things? Proper base pairing like this would require 61 different anticodons for the 61 different codons that aren't stop codons. And GG anything is a glycine codon. Wouldn't it be convenient if a single anticodon could pair with G, G, and any third base? Indeed, things like this are possible due to a trait called wobble, first discovered by Francis Crick, whose other work you may be familiar with by this point. Certain tRNA anticodons can pair with multiple codons. The bacterium Mycoplasma has only 30 different tRNA genes representing 29 different anticodons. E. coli has 79 tRNA genes, but they only represent 42 anticodons. Both are far short of the 61 different non-stop codons that must have tRNA partners. This is possible due to the flexibility of the third position of the anticodon. Because the anticodon is part of a loop and not strictly linear, the base in the 5' position of the anticodon, the wobble position, doesn't base pair as you normally expect. While G normally pairs with C, in the wobble position it can pair with C or U. So the tRNA anticodon, AAG, can pair with the codons UUC or UUU. A single anticodon can work with both potential phenylalanine codons. The table on the right shows some of the other wobble rules Crick determined. The use of modified bases in this position further expands the combinations of base pairing available to the cell. Putting the wobble rules and the codon table side by side can illustrate some of the usefulness. Many times a U or C in the third position codes for the same amino acid, so having G that can pair with both is handy. Likewise, many times A or G in the third position makes no difference, so it's good to have two thiouracil derivatives that can pair with both. Other times, the third base makes no difference at all. Now, there's no single base that's great at pairing with A, C, G, and U. There are 5 oxyuracil derivatives that come close, but aren't great at pairing with C. So maybe it takes 2 anticodons, that one and one with G in the third position. Or maybe an anticodon with C in the wobble position and another with inosine. There are several combinations and only require 2 anticodons for the 4 possible codons. Inosine by itself is interesting because it wobble pairs with U, C, and A. These are the 3 possible bases in the third position of isoleucine codons, so a single anticodon can cover all of those. Finally, there are cases where wobble pairing would be bad. A methionine tRNA should only pair with AUG, and a tryptophan tRNA should only pair with UGG. So it's good that both cytosine and 5-methylene uracil derivatives are specific in their pairing with G, even in the wobble position. That's what I want to cover for tRNAs. Next, we need to get into the actual process of protein synthesis. We have mRNA sequences that direct the order of amino acids, and we have tRNAs that base pair with the codons and bring in the corresponding amino acid. But how are the amino acids attached to the tRNAs brought together into a polypeptide chain? That's a pretty big topic, so let's cut this video short and cover that next time.